The way our brain puts an end to fear memories has long been a mystery, but recent scientific breakthroughs are finally unraveling these secrets, offering hope for those battling anxiety and trauma. But here's where it gets controversial. Fear extinction, the process by which we learn that something once scary is now safe, may be far trickier than we thought. And the environment where this learning happens plays a huge role in whether the fear truly stays gone. In a groundbreaking study, scientists have, for the first time, pinpointed the specific brain signals connected to how humans extinguish fear memories. Using cutting-edge techniques involving implanted electrodes combined with a sophisticated method called Representational Similarity Analysis, RSA, they discovered that when people learn that former threats are now safe, their brains show an increase in a kind of rhythmic activity known as theta oscillations. But crucially, this increase happens in the amygdala, the brain's emotional processing hub. What makes this discovery fascinating is that these extinction memories, the new safety memories created during therapy, are highly specific to the context in which they were formed. This sheds light on a frustrating problem many face: why fear often creeps back after therapy ends or outside the safe zone of treatment. It suggests that the brain treats the safe environment as a special, almost isolated event, instead of a change that generalizes everywhere. This context dependence is a key reason why fear relapse happens. The deeper understanding of these brain processes opens exciting new doors for treatments that could target fear-related disorders like post-traumatic stress disorder, PTSD, and anxiety, with greater precision and long-term effectiveness. Let's break down the key insights. Amygdala's role. Theta waves, patterns of brain waves oscillating at a certain frequency in the amygdala, ramp up when a previously fear-inducing signal is learned to be safe, acting like a neural all-clear. This signals the brain shift from danger to safety learning. Context is king. The new safety memories are not universal. They're tightly linked to the setting where extinction takes place. This means that if you leave that environment, the original fear can come roaring back, explaining why fear often reemerges in different contexts. Human confirmation. While animal studies had hinted at this system, this research provides the first direct proof it works the same way in humans, bridging a crucial gap between animal models and human brain function. Fear suppression is vital for healthy behavior because it helps us avoid overreacting to memories that could otherwise trigger anxiety or depression. Up until now, the dominant idea has been that extinction happens when the brain creates new memories that override the fear, but that these memories depend heavily on context. Much of what researchers knew came from rodent studies, where specific brain wave patterns between the amygdala and hippocampus were linked to fear learning and extinction. Yet, until this study, no one had confirmed if human brains show the same pattern. Published in Nature Human Behavior, the paper from Universitat Autònoma de Barcelona and Ruhr-Universitat Bochum finally crack this mystery. They used RSA, a powerful tool that goes beyond just measuring brain activation and lets scientists understand how neural representations, the brain's way of encoding specific memories, work in intricate detail. Daniel Pacheco Estefan, the lead author, explains that this technique lets researchers see the detailed mechanics of how episodic memories, memories of events, form and fade, offering a richer view than traditional methods. The experiment's design was especially innovative. It involved presenting multiple visual cues within varied contexts during phases of memory learning, extinction, and testing. This allowed the team to replicate classic conditioning studies, humans, validating theories mainly derived from animals. Participants were 49 epilepsy patients who already had electrodes implanted in brain regions involved in fear and extinction due to their medical treatment. These patients viewed neutral images like a hairdryer, a fan, and a toaster. Some images were paired with an unpleasant sound, creating a mild fear response, while others were not. Later, the images were shown again without the unpleasant sound to encourage extinction. Here's what they found. During extinction learning, when the unpleasant association was removed, the amygdala showed increased theta activity in response to former threat cues. This is interpreted as the brain signaling these cues are now safe. The brain patterns for items previously paired with negative sounds were more similar to each other, highlighting a common neural signature for fear-related memories. These extinction memories were tightly tied to the context where they were learned. When tested outside the original context, the old fear memories were more likely to resurface. Pacheco-Estefan emphasizes this could explain why extinguished fears tend to return once therapy stops. Because the brain sees the therapeutic environment as a unique, safe episode rather than a general rule, Nikolai Axmacher, the project's coordinator, adds that extinction memories might be stored much like other episodic memories, unique, specific events, suggesting the safe conditions learned during therapy might be perceived by the brain as rare exceptions. This sparks an important conversation. If our brains treat safety learning like rare exceptions tied to specific places, should therapies focus more on generalizing extinction across different contexts? Could this be why some therapies don't hold up long-term? Ultimately, these insights offer a promising foundation for developing better treatments that consider not just fear extinction itself, but the context dependency of that extinction. This could revolutionize how clinicians approach anxiety and trauma-related disorders, tailoring treatments to help fear memories truly fade across real-life situations. These pioneering findings reveal crucial physiological and representational brain processes behind fear extinction in humans, and encourage us to rethink how we can help patients achieve lasting relief from fear. What do you think? Does the brain's apparent context lock on safety memories mean we need a radical shift in therapy methods? Or can existing treatments be adapted to overcome this hurdle? Share your thoughts and join the conversation.